Okay, so now you know about the clinical samples and how the data derived from those is structured. So now let's get into the really exciting part, which is what you're going to be working on. What are the 3 crunches you're going to be kind of tackling here? So let's start with the first crunch. So here we're going to be asking you to predict gene expression from tissue pathology images. So your training data is going to be paired data where we give you the H&E image and the spatial transcriptomics data. And we're going to provide you 460 genes that were measured in the spatial transcriptomics data. And then in the test, we're going to hold out patches of tissue that are outlined here in a gray box. And in this patch, we're going to give you the H&E image, but we're going to hide from you the spatial transcriptomics data. And we're going to ask you to take that H&E image and predict this transcriptomics data for the cells that are in that tissue patch. So in this cartoon here, we have cells A through F, and we're going to ask you to predict the expression of gene 1, 2, 3, all the way to gene 460. And then we're going to see how well you do because we actually know the underlying ground truth because we've carried out the spatial transcriptomics experiment for this entire tissue. So again, starting with the H&E image, can you predict spatial transcriptomics data for all the cells in this held out tissue patch? So that's the first crunch. The second crunch, we make things more difficult for you in that we are going to ask you to predict the gene expression for genes that were never measured by the spatial transcriptomics experiment. So we've chosen 20 genes that were not measured by spatial transcriptomics, and we are going to ask you to predict their expression. So the structure of this is pretty similar to Crunch 1 in that we are going to provide you with paired data, and we're providing you with the H&E image and the spatial transcriptomics data for 460 genes. And we are going to hold out tissue patches, and from the H&E image, we're going to ask you to predict the held out spatial transcriptomics data, but in addition, 20 genes that were never measured by spatial transcriptomics. So you're not going to just be predicting genes 1 through 460, but also genes 461, 462, all the way to 480. And now you're asking, well, how would I know anything about those 20 genes? This is where all our background about single-cell transcriptomics comes in. We are going to provide you with single-cell transcriptomics data from ulcerative colitis patients, and that data has all 20,000 genes measured. So it has the 460 genes we provide you in spatial transcriptomics, it has the 20 genes that we would like you to predict, and it has all other 19,520 genes provided. So can you use the single-cell transcriptomics data to tell us what are the gene expression levels for those 20 genes that we're really interested in? So that's Crunch 2. It's really an out-of-distribution problem because we're never providing you spatial transcriptomics data for those 20 genes. Okay, so that's Crunches 1 and 2. And the final crunch is focused on detecting cancer earlier. So the focus is on this really special ulcerative colitis tissue section where there's both a non-cancerous region in the colon mucosa and this dysplasia region. And remember, dysplasia is this sort of abnormal growth of epithelial cells that's precancerous. And what we would like is to find genes that differentiate or that mark the dysplasia region from the non-cancerous region. So what are genes that are more highly expressed in this dysplasia region compared to the non-cancerous region? And these are genes that can maybe tell us what's going on in these early cancer events. So there are many ways to go about doing this, but if you've done Crunches 1 and 2, you will have built models that can take the H&E image and predict gene expression. And so one very reasonable way to proceed is to take the H&E image here and predict the gene expression in the different regions, in the dysplasia region and the non-cancerous region. And so we can make those gene expression predictions in the non-cancerous regions here for cells here, and in the dysplasia region for cells here, and get all these predicted gene expression vectors. And one thing I'll point out is that you actually have Xenium spatial transcriptomics data for the non-cancerous region, but we never provide you any Xenium spatial transcriptomics data for the dysplasia region. So for the left half of this tissue image, you're really predicting the genes beyond the 460 that have been measured already by spatial transcriptomics. But on the right half, you're really predicting all 20,000 genes. You do not have any spatial transcriptomics data directly from that piece of the tissue. And again, how do you predict genes that you were never, given any information about, we are going to provide you with single-cell transcriptomics, single-cell RNA sequencing data for dysplasia regions. So this is the data set that we had introduced earlier, and it's single-cell transcriptomics, and it's going to tell you what do epithelial cells with dysplasia look like in terms of their gene expression. So you can use that single-cell transcriptomics data to help with your predictions of what's going on with the dysplasia regions. And now once you've kind of made these predictions of gene expression in the non-cancerous region and gene expression in dysplasia regions, it's a relatively simple task to just compare the two and say what genes are marking this population of cells that are in the dysplasia region versus genes that are marking the non-cancerous region. So can you look at these and say what are the genes that distinguish these two regions once you've made these predictions? And so this is a pretty ambitious crunch. It's pretty exciting and cool. And one really exciting part of this too is that we're actually going to experimentally test your predictions. So when you predict a gene that marks dysplasia, we're actually going to go in and use that to design a new panel of 460 or 480 genes, and we're going to use that panel to measure a new piece of colon tissue that has dysplasia, and we're going to see if the genes that you predicted as marking dysplasia are really being expressed in the dysplasia region compared to the non-cancerous region. And so this is really an ultimate test. We're taking your predictions and applying them to a piece of tissue that we haven't measured before. So this is a really exciting opportunity. Most machine learning competitions, there's not really an experiment associated with. There's not really, something new that you're really doing. And here we actually have a chance to really go into patient specimens and see if we can really get better at predicting cancer in these tissue sections. Okay, great. So now we've gone through all 3 lectures in this crash course. Thanks for listening along. If you need more information, you can go to our website. We're really excited to see what you come up with as you tackle these crunches and to interact with you along the way. So good luck and have fun.